हेलो। हेलो।
नाही, फक्त जेव्हा मी डेकोरेशन करत होतो, तेव्हा तो नाही म्हणजे नाही.
हेलो।
हाँ हेलो।
गुड मॉर्निंग सर.
गुड मॉर्निंग रवंडे सर।
हाँ हाँ हेलो। हाँ सर।
स्टूडेंट ज्वाइन करे क्या?
हाँ वो 1 मिनट देख ही रहा हूँ।
सना
35
खान is there?
participants joined.
Yes, sir.
Okay. Other than Sana, who have joined among students?
फक्त जेव्हा मी डेकोरेशन करत होतो, तेव्हा तो नाही.
Sir Simran Patel.
हेलो।
सर अजना शबी.
हाँ हेलो।
गुड मॉर्निंग सर.
गुड मॉर्निंग रवंडे सर।
हाँ हाँ
स्टूडेंट ज्वाइन करे क्या?
हाँ वो 1 मिनट देख ही रहा हूँ।
सना खान is there?
Participants joined.
Yes, sir. Yes, sir.
Okay. Other than Sana, who have joined among students?
Azna Shabi. सर शालिनी चांदे मैडम की आईडी से हूँ मैं, अजना शबी.
Okay, okay,
Sir Simran Patel.
okay.
सर सेल्वी थोड़ी.
Okay, all of the participants, please keep ready your presentations. Every participant should get 7 minutes to present their oral poster or model. Okay. We will start the oral presentation from Sana Khan.
Okay, sir.
So, good morning to one and all. Okay. Today, we are going to start the oral poster and model presentations from 11 a.m. to 2 p.m. As we have some time constraints on the 6th of March. We have to finish our validatory program up to 4 p.m. So, and 7th will be the online mod day for the conference. And we have a number of resource person to discuss their innovative ideas on uh, topic of our conference. So we have decided to prepone the oral and poster presentation, and that we are conducting today only. So welcome one and all. Now I would like to invite Dr. Ashok Davande sir, convener of the conference, to preside over the presentations. Ashok Davande sir.
Thank you, sir. First of all, I would like to welcome all the presenter over here. I would like to give one brief introduction of tomorrow's conference on frontiers in life science and material science at AI and IPR interface. We have chose this topic because we know that the recent development in AI are bringing major changes in the field of life sciences and material science. AI helps researcher analyze large data, discover new materials, and improve research efficacy. Along with this scientific advancement, it is also important to understand how this intellectual property rights protect our new idea, invention, and innovation. The presentation explore how AI influencing the life sciences and material science, and how IPR play important role in protecting discoveries. Today we have number of presentation, oral and poster, and so we extend our best wishes to all students who are presenting their work. Hope this opportunity encourages them to share their ideas, build confidence, and contribute to scientific learning and innovation. Best of luck to all the participants. I request all the judges over here to start this presentation program. Thank you.
Thank you, Dr. Ashok Davande sir. Now, I would like to introduce our today's judges for the oral poster and model competition of the participants of national conference. Dr. Raju Gadpaile, Assistant Professor, Department of Microbiology, YC College, Lakhandur. is going to judge the today's oral poster and model presentation. Along with him, Dr. Mahesh Tengri, Department of Zoology, Assistant Professor and Head. Dr. Dhaneshwar Lanjewar, Department of Botany, Assistant Professor and Head. Dr. Bisen Sir, Assistant Professor and Head, Department of Physics, Yashwantrao Chauhan College, Lakhandur. Dr. Pranay Vasnik, Assistant Professor, Physics, Department of Physics, YC College, Lakhandur. Dr. Rokde Sir, Sumit Rokde Sir, Assistant Professor, Department of Physics. Dr. Nitesh Kamble Sir, Assistant Professor, Department of Botany, YC College, Lakhandur. Miss Sonam Bansod, Assistant Professor, Department of Zoology, YC College, Lakhandur. Dr. Snehal Zuare Madam from Department of Geology, YC College, Lakhandur. Dr. Gurde Sir, Assistant Professor, Department of Geology, YC College, Lakhandur. Dr. Maske Sir, Head Assistant Professor, Department of Chemistry, YC College, Lakhandur. Dr. Shailesh Bhagat Sir, Assistant Professor, Department of Chemistry, YC College, Lakhandur. Mr. Pankaj Ledade, Assistant Professor, Chemistry, YC College, Lakhandur. Mr. Kishore Itankar, Miss Neetu Parchakke Ma'am, Assistant Professor, Department of Chemistry. Other than this, we are having Dr. Mahesh Akolkar, Laboratory Technical Officer from Latur. Dr. Anil Narsinghe, Assistant Professor and Head Department of Microbiology from Yogeshwari Mahavidyalaya, Ambazogai. And Dr. Somnath Kirwale Sir, Assistant Professor and Head Department of Botany from Vaidyanath College, Parri, District Bid. And other juries are there to judge the today's presentation of the students. So I would like to welcome all the juries, all the referees to judge the oral poster and model presentation. So please, we continue with the oral and poster presentation, sir. Thank you. Now, I would like to hand over the this platform to all the juries. Dr. Gadpale, sir.
Thank you very much, sir. And
You may continue.
welcome to all the participants of National Conference. Dr. Banginwar, sir, is host for this particular oral and post presentation session. I welcome all the judges who are present over here for the judgment of the oral and poster presentations. And if the participants are ready, then we can call out to them for presentation, startup presentations. All the participants, they will have 7 minutes each. Before 7 minutes, there will be the warning for them to finish. If they exceed their time, it may be that the judges will decide whether to skip the questionnaire, because otherwise what will happen, they will take 7 minutes, and after that another presenter will be ready. So it's on the judgment of the judges. So it's better all the participants are requested to finish their presentation within time so that judges can ask them. So whether the presenters are ready with their presentations?
Yes, sir.
Yes, sir.
So, okay. I will ask Banginwar sir whether if he has the list of presenters who are ready for the
I have the
presentation?
list. I have the list, sir. The first
Okay.
one, Sana Pathan.
Sorry,
Yes.
one more thing, Doctor. I would like to welcome Dr. Yogesh Banginwar, Assistant Professor and Department of Microbiology from Pulgaon College. Sorry, Banginwar sir.
It's
मैं
okay,
नाम पे भूल
sir.
जाता हूँ. Okay, Sana Pathan. Sana Pathan,
Please
you may
share
start
your
your
slide.
presentation.
Please share your slide. Sana Pathan.
Hello.
Please share your screen.
Sir, my voice is audible.
Yes, yes.
My screen is visible.
No, your PPT is not coming.
Sir, why it is not coming?
Please open your PPT on your computer.
Sir, I have
And then-
opened.
Then share it. Next participant, Thul. Be ready with your presentation.
Uh, Sorry, sir, I am
Sana,
unable
please,
to share. I
Sana,
am
Sana,
sending my
please,
PPT to
Sana,
sir.
Sana,
Yes, sir.
minimize this screen, then PPT will visible. Minimize Zoom screen.
Now it is visible.
Blank.
Blank.
Try to change the slide.
Now. Sir, it's better. I will share my slide with you, PPT with you.
Okay.
Okay.
किसकी?
Sir, on National Conference 2026 WhatsApp group. I have shared, sir.
Yeah. मैं भेजा है अभी मेरे व्हाट्सएप पे उसने शेयर कर दिया। हाँ, एक मिनट अभी Just a minute. Now check it.
Yes, sir. Yes, sir. I can see my screen.
Yeah. So go ahead.
Okay, sir.
So 7 minutes.
Good morning. Yes, sir. Can I start, sir?
Yes.
Good morning to all the judges and participants. I, Sana Ayub Khan Pathan, research scholar from RTMNU Nagpur University. I'm presenting my research topic on antimicrobial resistant profile and beta-lactamase production in clinical isolates of Klebsiella pneumoniae from wound infection. My supervisor is Dr. N.B. Hirutkar sir from Department of Microbiology, Nabira Mahavidyalaya, Katol. My place of research work is Department of Molecular Biology and Genetic Engineering, RTMNU, Nagpur University. Next. So Klebsiella species, it is a gram-negative non-motile bacteria, and it is one of the most opportunistic human pathogen because it causes both the community and hospital-acquired infection. And it is resistant to many broad-spectrum beta-lactam antibiotics via many mechanisms, such as extended-spectrum beta-lactamase production, which is an increasing health concern. An infection due to this multi-drug resistant enterobacteriaceae is very important because it is a cause of morbidity and mortality worldwide. Next. Wound infection. This wound infection is the major cause of morbidity because this wound form a biofilm and it contains a bacterial bioburden. And if the integrity of this biofilm fail, the bacteria will not multiply and will not cause chronic infection. So it is very important to study from wound infection samples. And National Institute of Health also suggests that 80% of human infection disease is caused by biofilm. Next. So my aim and objective was my study aimed to identify Klebsiella pneumoniae isolated from clinical sample and to evaluate their antimicrobial resistant patterns along with the phenotypic detection of beta-lactamase enzyme, including extended spectrum beta-lactamase and ampicillin C beta-lactamase. Next. 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 My study period was from January to December 2025, and my subject was Klebsiella pneumoniae isolated from wound sample. So I have processed total 55 strain, out of which I have found 6 strain resistant. And this 55 Klebsiella pneumoniae was identified by using the standard microbiological technique, and AST was done as per the CLSI guideline. And the remaining, the 6 strains were resistant to carbapenem, and the remaining strains were susceptible. Those susceptible to carbapenem were tested for ESBL and ampicillin C by Dix approximation method. Next. So starting from the result gender-wise, so many isolates were from the male as compared to female, that is 56%. Next. Then the age group was elderly age group, that is 46 to 60, 14 and 31 to 40 age group, 13 isolates were isolated from the elderly age group. Next. And most of the isolates were from the surgical ward because after surgery the patient are more explosive and they required a more prolonged bed stay. So 41.8% isolates were from the surgical ward as compared to the medicine, orthopedic, obs, gynec, pediatrician, ENT. Next. So coming to antimicrobial resistant pattern, it was seen that the highest proportion of resistant was observed against cefazolin, that is 20%, and ceftriaxone, that is 18.80%. It indicates that there is a high level of resistance to this third generation cephalosporin. And then the moderate resistance was seen with amikacin, levofloxacin, with gentamicin, that is 14.55%, 12.73% levofloxacin, and 12.73% gentamicin. It reflects there is a reduced effectiveness of aminoglycosides and fluoroquinones. And generally, these antibiotics are used in wound infection. And lower resistance was observed in piperacillin, tazobactam, and meropenem. But the resistant 10.91% resistant to meropenem is also significant because this is the last resort of drug. Next. And then beta-lactamase production was done among this pus-derived Klebsiella pneumoniae isolate, and 41.8% was ESBL production among among the isolate, and which indicate there is a widespread of resistance to the third generation cephalosporin. And 29.1% was detected in ampicillin C beta-lactamase was detected in isolate, and this ampicillin C producer is also health concern, because this confirmed that there is a resistance to cefamycin, which is not inhibited by clavulanic acid. It makes the routine detection and treatment more challenging for the health facility. And 40% resistant was to carbapenem. Next. So my study collaborated with many study from India, that is isolation of Klebsiella pneumoniae from pus samples, and more of the samples were isolated from surgical ward. My study concordance with Patel Sachin et al. From India, and the high resistance to third generation cephalosporin was observed as compared to reported by Patel et al. In 2017 and Sharma et al. In 2019, which document the widespread ESBL mediated resistance in pus-derived Klebsiella pneumoniae isolate. Then ESBL production 41.8% was also predominant resistant mechanism, which is observed in 2016 and 18 study by Singh and Das et al., who reported ESBL as the most common beta-lactamase in pus isolate. And also the coexistence of ESBL, ampicillin C, and carbapenemase production corroborates these study findings from Mathur et al. In 2015, which emphasizes the growing complexity of antimicrobial resistant patient in tertiary care hospital. Next. So coming to conclusion, it is very important to study the antimicrobial resistant patterns of this Klebsiella pneumoniae from wound samples, because there is a high prevalence of MDR, as Klebsiella pneumoniae is one of the opportunistic pathogen and it causes an hospital acquired infection. So there need of a routine resistant monitoring, infection control, and antibiotic stewardship for the effective management. Thank you. Next.
Okay, nice presentation.
Thank you, sir.
I have one question. You said that in the presentation that these organisms that you have isolated are resistant to which particular bacteria group of antibiotics?
Cefazolin, third generation cephalosporin, sir.
Okay, so these are basically fungal produced antibiotics, right? Cephalosporin and cephalosporin.
Yes.
So, what do you think could be the mechanism of resistance or sensitivity towards these antibiotics?
Actually, sir, resistance to these antibiotics suggest a widespread of beta-lactamase mediated resistance among these isolates.
So, you consider that beta-lactamase production is required for the or essential for resistance against the different type of category of antibiotics, because cephalosporins, they are not beta-lactamase.
Yes, they are not.
So
but
what you
sir,
can...
they are third generation antibiotic now.
Okay.
That's why.
Any question from any other judges?
So which media are you using for the isolation of the Klebsiella pneumoniae?
Sir, blood agar.
It is sufficient? Only blood agar?
Blood agar, sir, McConkey agar.
Which type of colonies are seen on the blood agar?
Sir, blood agar beta. On blood agar, sir, hemolytic colonies.
It is sufficient for the identification?
Sir, I have done the that IMBIC method also, and
but you are not mentioning in your slides.
Yes, sir, I have not mentioned that in my slide.
Okay.
Any other?
Any other judges?
Then we can go to next participant.
Yes, Thul, be
Thank
ready
you,
with
sir.
your presentation.
Hello, sir.
Yes,
So can I share my screen?
yes, definitely you can. बस तैयारी है बस मेरा हो गया इधर.
Hello, sir.
Yes. Go ahead.
Good morning, everyone. Myself, Sedvi Thool, research scholar from Department of Biochemistry, Dr. Ambedkar College, Nagpur. My topic of presentation is green synthesis, characterization, and antimicrobial evaluation of silver nanoparticles derived from Calotropis gigantea leaf. Content, introduction, material and method, procedure of synthesis, characterization, and antimicrobial activity. So, what are nanoparticles? The term nano is derived from the Greek word meaning dwarf or extremely small. These particles are tiny material exist from the scale 1 to 100 nanometers. They are being classified based on their origin, properties, and structures, such as one-dimensional, two-dimensional, and three-dimensional. So, how are these particles made? So, there are two major methods to synthesize nanoparticles, that is top-down and bottom-up approach. There are various methods to synthesize nanoparticles, such as electrochemical synthesis, sputtering, ball marrying, chemical synthesis, and green synthesis. Nowadays, the development of green method of synthesis has become a major focus for researchers, as it is a clear, non-toxic, cost-efficient, and environment-free approach. But why we are using nanoparticles? Due to their high surface area to volume ratio, it makes it in a unique it allows it to be in various fields such as chemical, food, electrical, healthcare, industry. Plant is one of the rich source as a stabilizing and caping agent. Some of the most assured nanoparticles are gold, zinc, platinum, copper, and silver. So here is the process of synthesis. Silver nanoparticles. The Calotropis gigantea leaf were collected from the local region of Nagpur. The leaves were washed thoroughly with distilled water. Silver nitrate was used as a stock solution for silver nanoparticles, and distilled water was used as a solvent. The mixture solution was incubated for 24 hours and further centrifuged and washed thoroughly. The finally synthesized nanoparticles were collected and stored in a airtight condition. So to confirm the formation of nanoparticles, we have characterized the nanoparticles with XRD. As you can see, the highest peak at 111 plane 11 at 2 theta degree with 32.30 degree, and rest are the other sharp peaks, which confirms the crystalline structure of the nanoparticles. Here is the antimicrobial activity on E. coli and S. aureus, that is gram-positive and gram-negative bacteria. Various concentration were taken as 50, 100, and 150 microgram per liter. The highest zone of inhibition was observed at 150 microliter for E. coli and S. aureus, and it was compared with ampicillin, and the negative control was DMSO. Thank you.
Okay. So anyone question?
Hello, sir.
Yeah.
I
Proceed,
have
sir.
2 or 3 queries regarding this topic. I want to ask.
Yes, sir.
You performed the XRD, but you didn't told us about the size of the silver nanoparticle which you are synthesized.
Yes,
So
sir.
can you?
I have also characterized it with the SEM and TEM also, but I haven't attached it in the slide.
Okay, no, that's not the issue. No, I don't want same or tame analysis, but I want the particle size with respect to
But the silver
XRD analysis.
nanoparticles are at the size between 30 to 40 nanometers.
30 to 40. What is the average size? You can calculate from the given XRD data only.
Yes, sir. It is in the range of 30 to 40.
Okay, so my next question is, at what temperature you prepared the silver nanoparticle?
Sir, it was at room temperature.
What?
Room temperature.
Room temperature, but you mentioned that you are performing oven heating. So at what
Sir,
temperature?
that was for heating, like
At
for the
what
liquid
temperature?
to solid. From liquid to solid, I have to solidify it. So I have heated my nanoparticles in a hot air oven at 80 to 90 degrees Celsius.
80 to 90.
Yes, sir.
So it is for the only for the dryness.
Yes, sir.
Okay. The next question is regarding the IR spectra. You didn't mention there because
Okay, sir.
IR gives the idea about the what are the approximate capping agent, reducing agents are there. So you are not mentioned there. Where is the IR data?
Okay, sir.
Okay, you performed very well method and you prepare silver nanoparticle. That's good. Thank you so much, sir.
So
Thank you, sir.
one question from my side. Thul.
Yes, sir.
So you are using the ampicillin
Yes, sir. As a positive control.
for E. coli.
E. coli and S. aureus. Yes, sir.
But the E. coli is the gram-negative microorganism, and ampicillin it works against the gram-positive. How it is possible? As a
Okay, sir.
microbiologist, E. coli is a gram-negative.
Yes, sir.
E. coli do not having the any type of the particularly the cell wall, tough cell wall structure.
Okay,
And
sir.
you are using the ampicillin. It is work against the gram-positive. That will be the beta-lactamase producers.
Okay, sir.
Okay. So next participant,
Any other
Patel.
questions? If there are no questions, we can move ahead.
Next one,
Next participant, be ready.
Simran Patel.
Yes, sir. So is my screen visible?
Yes, yes.
Am I audible, sir? Hello?
Yes,
Continue,
please go
continue.
ahead with your presentation.
Okay. Hello, good afternoon, everyone. My name is Simran Patel. I am pursuing PhD at Dr. Ambedkar College under the supervision of Dr. Bhuvaneshwari Mehere, Professor, Dr. Ambedkar College, Nagpur. My topic is development and characterization of vinblastine-loaded phospholipid-based phytosome. As we all know that cancer is the second leading cause of death globally after ischemic heart diseases, as cancer cells proliferate quickly, which causes them to grow and simultaneously infiltrate neighboring cells. During cancer treatment, Conventional therapy, like immunotherapy, surgery, radiotherapy, often harm healthy cells, which can have fatal consequences. To cure this problem, an Italian pharmaceutical company, Indena, developed the first phytosome in late 1980s. Phytosomes are a patented drug delivery system designed to enhance the bioavailability of absorption of natural plant extracts by complexing them with phospholipids, that is phosphatidylcholine. It aims to improve the bioavailability of the drug by targeted release of the drug to the targeted location.
सर मागच्या वर्षी आपण डायग्नोस्टिक टेस्ट घेतली होती ना.
These are the current phytosomal therapy against
screen
various
share करा ना.
cancers. Lutein is used to treat colorectal cancer. Curcumin is
हाँ, हाँ,
used to
ते-
treat breast cancer.
हो, हमने इनको
Placidaxel and gemcitabine is used to treat pancreatic cancer. Vincristine is used to treat brain cancer. Uh,
आज आता आपण
Resveratrol is used to treat cervical cancer, and so on. Here, I have taken the drug vinblastine. There's a research gap found on the drug vinblastine. Vinblastine is a potent, naturally derived alkaloid chemotherapy medication isolated from the Madagascar periwinkle plant, Catharus roseus. It is used to treat breast cancer, testicular cancer, Hodgkin lymphoma, non-Hodgkin lymphoma, and cervical cancer. The mechanism of the drug vinblastine is vinblastine occurs as the cell cycle in metaphase, causing mitotic inhibition, and then it binds specifically to tubulin and block their polymerization to form microtubules. Due to lack of an intact mitotic spindle, the chromosome disperse, and which leads to cell death. My first objective is to synthesize vinblastine based phytosome using two methods: solvent evaporation method and anti-solvent precipitation method. Second, to characterize a newly synthesized phytosome using various analytical techniques: UV visible spectroscopy, FTIR, XRD, SEM, TEM, etc. Third, to detect the in vitro anti-cancer potential of newly synthesized phytosomes. And fourth, to study the comparative efficacy of newly synthesized phytosome drug or phytosome over naked vinblastine drug. Here, I have taken the drug vinblastine and it is mixed with the phosphatidylcholine and dichloromethane, which is an organic solvent. It is then transferred to round bottom flask and kept in and refluxed in totally evaporated at 40 degrees Celsius for 2 hours. After 2 hours, the solvent was evaporated and we got the thin film. The thin film was obtained at the bottom of the flask. This thin film was scraped and transferred in beaker with N-hexane. N-hexane is an precipitator. The mixture was then filtered and transferred in Petri plate for complete drying in desiccator for 24 hours. After 24 hours, the sample was dried and crushed, powdered by the help of pestle model and transferred in Eppendorf tube for further use. This is the preliminary evaluation of the phytosome formation is done by the help of UV visible spectroscopy. Here, it can be seen there are two characteristic peaks, absorption peaks can be seen at 218 nanometer and at 272 nanometer. This is the XRD analysis. This XRD analysis suggests revealed that it has less crystalline structures have been formed. The size of the particle, the average size of the particle formed is 37.95 nanometer. This is the FTI analysis. Here, here there is a hydroxyl bond can be seen at 3304.46 nanometer, which confirms the formation of a drug and phospholipid complex. This second shift can be seen at 1224.53 nanometer, which suggests the formation of phosphate bond. The combined data of FTR and of XRD and FTR suggest that the drug has been successfully incorporated and it's likely to have better solubility and dissolve more easily and be absorbed more effectively by the body. These are the references. Thank you.
Any questions? So from my side, one question.
Yes, sir.
You are using already the vinblastine.
Yes, sir.
It is in a pure form.
Uh, uh, yes, sir, it is in pure form, but it contains preservative like NaCl.
Okay. Are you using any cancerous cell line?
Yes, sir. I'm using MCF7, but I am at my first objective to synthesize. I am using cancer cell line MCF7 and HepG2. It is my third objective.
Okay.
Okay, I have a question.
Yes, sir.
What was the concentration of vinblastine ratio to the phospholipid that you have used?
Sir, is 1:1 ratio.
Okay.
One part of the drug and one part of the phosphatidylcholine is 0.0011 millimoles of drug and 0.0011 millimoles of phosphatidylcholine.
And how did you prepare the phytosomes?
So I have prepared by the method of solvent evaporation.
Solvent evaporation.
Yes, sir.
And did you see the structures formed by any other method?
So I have currently I have done XRD and FTIR. My sample is I have sent my samples for cement
No.
analysis, and the results is yet to be come.
I'm asking, how did you suspended these two? What was the mixing method you have used?
Okay. In this dichloromethane, which is an organic solvent, is used, and which is then refluxed in the rotary evaporator for complete drying.
That is for drying.
Yeah.
I'm saying for loading of vinblastine, okay, for mycelium formation, there should be some technique, right? So what technique you have used for, vinblastine loading as a phytosome?
So sorry, I'm not understanding the question.
Okay, my question is, I'm rephrasing it.
Yeah.
You have mentioned that vinblastine loading, right, in the phospholipid-based phytosome.
Yes,
So
sir.
there should be, formation of phytosomes. So you have to
Yes.
ensure whether phytosomes are formed. That has to be seen visually
Yes,
or by
sir.
some technique. Okay. Or
Yes,
you
sir.
can do some spectrophotometric analysis. Second is, what is the concentration of vinblastine loaded per phytosomal mycelium formations, so that you can go for characterization.
Okay, sir. So I have to read
Okay.
about it.
Okay.
Okay.
All
Thank
the
you, sir.
best.
Thank you, sir.
Any other question from JS? Okay, thank you. Next participant, please be ready.
Please ready. Dr. Bhavna Mohite, Assistant Professor from Bajaj College of Science, Wardha. Be ready with your presentation, ma'am. Yes, continue, madam. Mohite, madam.
Yes, sir.
Continue, madam, with your presentation.
Yes, sir. So is the screen is visible?
Yes, yes.
Yes. Thank you so much, sir. So good morning, everyone. I'm Bhavna Mohite, from the Department of Microbiology, Bajaj College of Science, Wardha. Today, it's my pleasure to present part of my work regarding probiotics, that how it shifts from the just not only as a gut guardian, but it could be a good biotherapeutic agent. So we all know that antibiotic resistance is a rising problem towards the various common pathogens that we have. And so that's why there is a need of some safe and natural type of antimicrobial alternative. So we all know regarding the role of probiotics that it going to work to maintain our good microbiota. But at the same time, it has some of the characteristics, like it's going to inhibit the pathogens as well due to its various types of metabolite, including some acids or bacteriocins. And so we could use this probiotics, the biotherapeutic agent, further. And when we talk about these strains, which works as a probiotic organism, as the probiotic characteristic, it's having the important properties like showing this tolerance towards the acid or bile salt. And so that's why we could further have its use further in as a biotherapeutic agents. So by keeping this in mind, we set some objectives. We started with the isolation of this lactic acid bacteria, particularly from the various fermented food. Then checking for its various probiotic traits. Then with its biosafety assessment, of course, and antimicrobial activity screening. And then further, after the potent organism among the isolates, we confirm with its identity at molecular level. And then we tried at lab scale that can we go with some capsule formation, which could be further, of course, it needs to be having the various criteria to be followed to be use it as a capsule or as a drug further, we could say. But it is at the experimental trial. So this is our methodology. We starts with collecting the various samples from fermented food. Then we isolated particularly we are targeting for the probiotic strain by using the selective media like MRS agar. Then the potent isolates, what we get, we try to all the isolates get confirmed at the morphological and biochemical characteristic at the lab scale. And the potent one we identified with molecular technique. Then with some safety assessment we have done, and we check for its antimicrobial activity. And further, regarding that, its antimicrobial activity and further its stability and that type of study and viability study were carried out. So it starts with the various isolates. So out of that most potent, what we found among the 10 isolates we have taken and compare it on the basis further that which one is need to be more potent among the all. So we found that among the all isolates, the one of the isolate, it is going to be the one which is giving the characteristic of all probiotic traits like the acid tolerance, bile salt tolerance, and the temperature. So we found that the potent one, we identified it at the lab scale, which is gram-positive rods or cocci. Among that, different types of isolates, which are catalates. Then it is RNase negative non-spore former, and showing the non-hemolytic activity. So this we will further we use. Among that, the most potent we confirm with type of molecular characterization. So these are the various isolates. We further move on from the 10 to further confirm, and these based on their probiotic characteristic, it was checked with its probiotic trait whether it is actually or not. Then another parameters, like apart from this acid tolerance, bile salt tolerance, sodium chloride tolerance, temperature tolerance, the antibiotic sensitivity we have checked for this. We found some of them like been showing the sensitivity pattern for most of them. Along with them, some of them showing the some intermediate one. Then we check its antimicrobial activity against the most common type of pathogens, including Staphylococcus aureus, Escherichia coli, Salmonella typhi, and Pseudomonas aeruginosa. Then after that, the potent isolate that we found based upon this trait of probiotics and of course the antimicrobial activity, we go for identification of that potent isolate by 16S rRNA sequencing and found to be that it is Lactobacillus plantarum. Then after this, as this organism is having the antimicrobial activity as well against the various common gram-positive and gram-negative pathogens. So at lab scale, we tried to go for its probiotic capsule formation with this freeze-dried probiotic biomass, and further we going to encapsulate it in the form of capsules. We check it's like this is the composition like with some lyoprotectants and within the shell of gelatin, which could be work for the oral delivery. And of course, it's having some prebiotics content as well, which will going to support for it. But this is, we could not say that at the level, but we just try it at the laboratory scale that whether we could go with this or not. So what the capsules were prepared, we check for its viability and stability over a period of months. That it found that it retained the activity, but it's not there that the 100% of the activity is going to maintain with its viability. We found that over the period of time, it's get reduced. So we found that it needs to have there is a need to have some improvement with respect to the viability and stability. So we are carrying out this study presently as well. So to maintain its viability for a longer duration of time. So it is why we go with this probiotic one to be used as a biotherapeutic agent. So it is just given a comparison that what we have at present, and with respect to that, what is the cost distribution per probiotic capsule will be there. So we found that it's comparatively kind of a cost-effective one. So it's been a proposed idea that if you go with like this way, that probiotic and with its biotherapeutic one, so it could be a kind of novel idea, novel in the sense that it's been there that the various this idea has been proposed, but at present also in the market, we do not have such type of probiotic culture for this purpose. So it means, of course, it's having this societal and health impact of it. So we conclude our study with this, that we could use the probiotic type of culture, showing the probiotic traits and having the antimicrobial activity potential. And by checking its biosafety and all, like we could further use it for the purpose of like as a biotherapeutic agents. So these are some of references, and I'm lastly, thank you for giving this opportunity to share some of the primary work here. Thank you so much.
Now, house is open. Ask the questions.
Thank you, madam. It was nice work to share. I have one question.
Yes.
We basically see any organism as a probiotic that basically delivers some of the characteristics, and one of the characteristics you have showed is the bile tolerance, temperature. I think it was not temperature, bile tolerance was there, and antimicrobial activity against one of the pathogenic bacteria. Can you suggest there are any other parameters that we can study for these organisms as a probiotic organism?
See, sir, these are the what I will mention over here. Apart from that, there are various probiotic traits that we should need to be maintained. Like first one is that in the body, it is been in consortium with the other organism. So we need to check that how it will be going to affect the other bacteria which are being present. The next one is like mucin binding type of assay we can do. Along with that, this is primarily we are done with the hemolytic assay, but the other biosafety characteristics that also need to be checked.
Sure, right. Because some of these organisms, they produce some toxic elements that destroy the microflora of the gut, and therefore
that's why we need to be first confirm with its safety, biosafety. Yes.
Right. Thank you very much, ma'am.
Thank you so
Any
much.
other questions? I also have one question.
Yes, sir.
So regarding
Please.
the bioformulation that
Yes,
for
sir.
making the capsule. So what
Yes,
was
sir.
the
Yes, sir.
components that you have added to make the powder and refill the capsule?
Sir, we did taking the biomass and then lyophilize it. And with that lyophilized powder, we added the lyoprotectants,
Okay.
like trehalose sugar and then it is being get within the gelatin capsule. It is being done.
Okay.
So that's why
Thank-
sir, the very important thing
Right,
is regarding the viability as well. That needs to be
Right.
further, should be
Yes, yes.
longer lasting.
Right.
Yes.
Thank you.
Thank you so much.
Yes, next participant, Pooja Chauhan.
Thank you, sir.
Yes, sir.
Yeah. Please share your PPT.
Yes, sir. Sir, is it visible?
Yes, sir. Yes,
Okay.
yes.
Okay.
In a full size.
Size. Sir, is it visible now?
Yeah.
Okay. Good morning, all of you. I am Pooja Balwant Chauhan, PhD student of Dr. Bhuneshwari Mere ma'am from Department of Biochemistry, Dr. Ambedkar College, Diksha Bhumi, Nagpur. Today, I am going to give presentation on green synthesis and comprehensive physiochemical characterization of selenium nanoparticle using Jatropha curcus leaf extract. This is my introduction. Nanotechnology. Nanotechnology deals with the synthesis and application of material at the nanoscale, that is 1 to 100 nanometer in size, where the material exhibit unique physical, chemical and biological properties. I have selected selenium because selenium is an essential micronutrient with antioxidant, anti-cancer and antimicrobial properties. But bulk selenium exhibit higher toxicity and limited bioavailability. When we convert the selenium into selenium nanoparticle overcome these limitation of high toxicity and bioavailability, overcome this limitation due to their biocompatibility and advanced bioavailability, resulting in reduced toxicity and better biological activity. There are various methods of synthesizing nanoparticle, like physical method, chemical method, but these methods are harmful and environmentally unsafe. So, green synthesis method using especially plant extract is environmental friendly, that is eco-friendly and cost-effective alternative. So the plant which are selected is Jatropha curcus. Jatropha curcus leaf extract contain bioactive compound that act as a reducing and stabilizing agent. This study focused on the green synthesis and characterization of selenium nanoparticle using Jatropha curcus. Jatropha curcus, this is a picture of Jatropha curcus leaves. Jatropha curcus plant is of Euphorbia family. It contains phytochemical like alkaloid, glycoside, flavonoid, tannins, phenolics, saponins, terpenoids, and steroid, etc. They act as a reducing agent which convert metal ions into bionanoparticles, and it also acts as a capping agent which stabilize bionanoparticles and preventing their agglomeration. This is the materials and methodology. Synthesis of selenium nanoparticle. First, I have collected the fresh leaves of Jatropha curcus, shed dried it and for 2 to 3 days under shade. Then I coarsely ground the leaves. Then for preparation of extract, I have taken 5 gram of dried powder of Jatropha curcus, add to 100 ml distilled water, heat at 60 degree for 30 minutes. Then filter the extract with filter paper. Filtration of extract with, then again I filter it with Whatman filter paper 1, centrifuge the extract at 3000 RPM for 10 minutes. Then Jatropha, the product I get is Jatropha leaf extract. This Jatropha leaf extract, I use this Jatropha leaf extract for synthesis of selenium nanoparticle. For this, I add this extract with 30 millimolar sodium selenite anhydrous solution. When I add it, the color of the sodium selenite anhydrous solution changes to light brown in color. Then I heat at 60 degrees centigrade, heat the solution at 60 degrees centigrade for 1 hour on magnetic stirrer. After 28 hours, the color changes to ruby red after incubation of 24 hours. Centrifuge, then I centrifuge it, centrifuge the solution for 10 minutes at 11,000 RPM. The obtained pellet which I get, I collected it into the Petri plate. Then the obtained pellet were dried overnight in an hot air oven at 60 degrees centigrade. Nanoparticles were crushed into mortar and pestle. Then I collected this synthesized nanoparticle into the Eppendorf tube. Then I send the nanoparticles, selenium nanoparticles in for characterization. For characterization, there are various techniques, but I have go for optical analysis, morphological analysis, and structure analysis. Optical analysis, UV visible spectroscopy, which determines the optical properties and concentration of nanoparticle. For morphological analysis, transmission electron microscopy TEM and scanning electron microscopy SEM. TEM provides information on the size and shape at the atomic level, whereas SEM examines the surface morphology and topography. For structure analysis, I have sent the particle for X-ray, XRD, X-ray diffraction, and Fourier transform infrared spectroscopy, FTIR. X-ray identified the crystalline nature, whereas FTIR identified the functional group and chemical bond, which are responsible for forming nanoparticles. These are the results which I got. The characterization of selenium nanoparticle, the UV spectra of Jatropha curcus leaf extract, only leaf extract, which I got. I got a peak around 250 nanometer, and when I synthesize after synthesizing selenium nanoparticle from Jatropha curcus leaf extract, the peak I got is 397 nanometer of selenium nanoparticles. These are the X-ray diffraction report of Jatropha curcus selenium nanoparticle, where the X-ray XRD pattern of Jatropha curcus and selenium nanoparticle show prominent peak at 23.63 degree, 29.84 degree, and 43.78 degree, 51.84 degree, 61.66 degree, corresponding to 111, 200, 220, and 311 and 222 plane of face-centered cubic selenium, confirming the successful formation of selenium nanoparticle. The crystalline size calculated using the Debye-Scherrer equation range from 9.4 to 20.1 nanometer, with the average size of approximately 14.8 nanometer. These are the EDS spectra of Jatropha curcus selenium nanoparticle. Here, the metal selenium is clearly visible. This is the elemental spectra. Here we clearly see that the selenium element is here. These are the SEM analysis of Jatropha curcus SEM analysis report and TEM analysis report. Here, by the SEM analysis report reveals that the selenium nanoparticles are irregular and spherical in shape, whereas the TEM analysis of Jatropha curcus selenium nanoparticle reveals that the particles are formed with the range of 10 nanometer to 20 nanometer in size. And the conclusion of my study is Jatropha curcus leaf extract efficiently synthesized selenium nanoparticle, visual color change confirmed nanoparticle formation, characterization technique verified nanoscale particles and stability, green synthesis method is eco-friendly and cost-effective, synthesized selenium nanoparticles show promising application in nanotechnology and biotechnology. These are my references.
Stop, madam.
Thank you, sir.
So, any questions from the judges?
Okay, it was the nice presentation.
Thank you, sir.
So in the first slide, you mentioned that the aim for the production of selenium nanoparticles
Yes, sir.
was for bioavailability, right?
Yes, sir.
So what is the average range of selenium required for functional, physiology of an adult male or female?
Sir, please pardon, sir.
Okay. What is the concentration of selenium required in daily, diet?
Sir, actually, I need to read it.
Okay. No issue. Because selenium also has, toxicity,
Yes, sir.
right? So your aim, because you have mentioned it, your
Yes,
aim
yes, sir.
is to reduce the biotoxicity, right?
Yes, sir.
So therefore, you should have the background knowledge of what would be the range of concentration that can be given through
Yes.
the nanoparticles,
Okay.
isn't it?
Yes, sir. Thank you so much, sir. I will definitely read it.
Sure. And your presentation was nice because you have systematically presented the synthesis of nanoparticles. So have you characterized what are the capping molecules on formation of nanoparticles?
Yes, sir. Yes, sir. Sir, these are the FTIR interpretation of Jatropha selenium nanoparticle. Here, the aliphatic stretch and aromatic stretch. These are FTIR spectrum confirm that the phytochemical present in the plant extracts such as phenolic compound and flavonoids are involved in the reduction stabilization of selenium nanoparticle. This is the evidence of strengthened at 29.14 centimeter aromatic carboxylate vibration and strong COS stretch bond, while peak at 952 centimeter correspond to CH bending of aromatic and alkene, indicating the role of plant biomechanical as reducing and capping agents.
Okay. Thanks for sharing the data.
Thank you, sir.
Another question is
Yes.
to see.
So, sir, from my side, one question.
Yes, sir. Yes.
Pooja,
Yes,
what is
sir.
the utility of the Jatropha plant?
Sir, utilities, it is mainly for insecticidal activity, antimicrobial activity, anti-cancer activity. For anti-cancer activity study is going on, sir, and my topic is insecticidal activity to study the Jatropha selenium nanoparticle for insecticidal activity and antimicrobial activity, as Jatropha curcus itself possesses insecticidal activity and antimicrobial activity.
Which microorganism?
Sir, I am working on antifungal activity for antifungal activity on fluconazole. Fluconazole I am using as a control, and I am using.
Which diseases are causing in the human being, so that we can use the Jatropha selenium nanoparticle to get controls?
Sorry, sir, I need to read it for it.
Okay. No issue. So next participant, be ready, Rakshit. Please share your presentation. Thank you, madam.
Thank you, sir.
For your nice presentation.
Thank you, sir.
Sir, I have a question. Can we leave the meeting as we are done with our PPT?
Yes, sir. Definitely.
Okay, thank you, sir.
So next participant.
Yes, sir.
Rakshit, please share your presentation.
Sir, can I just share my presentation on the group?
Why?
If you can directly present it, please try to present it from your device. If there is any technical difficulty, then only you can share.
Yes, sir. So I'm sharing in the group, sir. There's a technical issue. Sir, I've shared in the group, sir.
Okay, just wait a minute. Yes, go ahead.
Okay. Good afternoon, everyone. Myself, Dhruhin Rakshit, a UG student of Department of Microbiology from North Bengal St. Xavier's College. So today I'm gonna present a poster upon the topic plucking out plucking out pollution, turning chicken feathers into organic fertilizer. So is my voice audible?
Yeah. Continue.
Okay. Okay. So chicken feather constitutes over 90% proteins, where the main component is keratin. Keratin is rigid in nature and cannot be degraded ideally. Chicken feathers mainly contains beta keratin, which is composed of beta pleated sheets, making it much stronger and brittle. Biologically, keratin is degraded by the action of keratinase enzymes produced by the keratinolytic bacteria like Bacillus species. These help in the breakdown of keratin by undergoing hydrolysis, which further includes sulfitolysis and proteolysis. So, could you just take up the slide into the methods part?
Yeah.
Okay. So in the methods, so the basic transformation of the chicken feather waste into the biofertilizer involves a microbial degradation. Yes, that table part. Into the microbial degradation utilizing the keratinolytic microorganisms. Given in my poster, there is a table of a keratinolytic microbes. These microbes produce the keratinase enzymes that hydrolyze keratin, resulting in nutrient-rich hydrolysis. These keratinolytic microorganisms produce enzymes, disulfide reductase and keratinase, which is induced in the presence of keratinous substrate. They have been found to completely solubilize feathers at a neutral to alkaline conditions. Most of these keratinolytic microbes are mesophiles, and they show a keratin degradation at 25 to 37 degrees Celsius. Some of them could exceed this optimal temperature. And finally, I'll go to the result part. In my poster there is two graphs present. So these two graphs shows that after the complete hydrolysis uh, of the following results, it was observed that these keratin consists of amino acids which are rich in macro and micronutrients. These macro and micronutrients helps in promoting the plant growth, and it makes and hence it becomes a eco-friendly fertilizer. Lastly, I would like to conclude by saying, sir, could you please take it down at the discussion part? Yeah. Lastly, I would like to Uh, just a second. Yeah. Yeah. So lastly, I would just like to conclude by saying that the chicken feather degradation is an eco-friendly and sustainable and economically beneficial solution in managing the poultry waste. Despite its advantages, it faces many challenges like slow keratin degradation and pathogen risks. To overcome these challenges, one must be aware of the following: that the bacteria must be more efficient and the sterility should be maintained. Awareness and policy support also encourages farmers and poultry producers to adopt this practice on a larger scale, replacing chemical fertilizers in the future. With proper investment and management, these organic fertilizers can contribute in the sustainable agriculture and resource effective farming. Thank you, sir.
So any questions from the jury members? Oh, I have one question.
Yes, sir.
What is the role of the bacteria to degrade
Role of
at 100 degrees Celsius? So that will be the 37 degrees Celsius. It is normally there, the Kirenolytica process. It is normally go in the soil at 37 degrees Celsius.
the bacteria like any keratinolytic bacteria, sir.
Okay.
Hello.
Can you using this as in the manure later on?
Sir, could you please repeat your question?
1 minute. Can you use as in the particularly the fertilizer for the agriculture land?
Yes, sir, we can.
Okay. Thank you.
Thank you, sir.
So next, the participant for the poster presentation, a car from North Bengal, St. Xavier College. Be ready with your poster presentation.
Sir, I have shared the PDF.
Yeah.
Poster. Sir, we are doing jointly, I and Shamika Bashar. Excuse me, sir. Hello.
Har, please continue.
Sir, since we are doing jointly, the first half of the poster will be covered by Shamika.
Go ahead.
Good morning, sir. Am I audible?
Yeah.
Okay, sir. Good morning, everyone present here. My name is Shamika Boshak, along with my friend Vichi Kaur. Today, we are presenting a poster on phage therapy, a new era in combating superbug. We are currently facing a major global health crisis known as antibiotic resistance. Antibiotic resistance occurs when bacteria survive antibiotic treatment and continue to multiply. These drug-resistant bacteria are often called superbugs, such as MRSA, that is methicillin-resistant Staphylococcus aureus, and VRE, that is vancomycin-resistant Enterococci. According to global reports, antimicrobial resistance was linked to nearly 5 million deaths in 2019. Beyond health, it also poses a huge economic burden, potentially costing trillions of dollars in the future. It affects major medical procedures like surgeries, chemotherapy, and organ transplant. So, the main question is, what is the solution? One promising alternative is phage therapy. Phage therapy uses bacteriophages or simply phages, which are viruses that specifically infect and kill bacteria. Unlike antibiotics, phages are highly specific. They target only particular bacteria without harming beneficial microbiota. Now, let's understand the mechanism. When phages are administered into the body, they attach to specific receptors on the surface of bacterial cells. After attachment, they inject their genetic material into the bacteria. Inside the bacterial cell, the phage multiplies using the host machinery. Eventually, the bacterial cell bursts, a process that is called lysis, releasing new phages that infect surrounding bacteria. This cycle continues until the bacterial population is reduced.
Historically, phage therapy was first discovered in the early 20th century by Felix de Herel. Although it lost popularity after antibiotics were discovered, interest has resurged due to increasing antibiotic resistance. Today, research is expanding globally, including advances in engineered and synthetic phages. Here are some modes through which engineered phages can be used as antibacterial agents. They can be used in phage therapy as well as phages-based endolysins, alteration gut microbiome, disrupting biofilms, altering antibiotic sensitivity, and delivery of antimicrobial. Now we move to the modifications in phage therapy. One important development in phage therapy is the combination therapy, where phages are used along with the antibiotics. This emerges as a promising strategy to combat antibiotic-resistant bacterial infections. Another use is of phage antibiotic synergy, which refers to the phenomenon where sublethal concentrations of certain antibiotics enhance the release of progeny phages from bacterial cells. The mechanisms of the synergy offer several advantages, such as enhanced bacterial killing, reduced development of resistance, effective action against biofilms. Biofilms are protective bacterial communities that are usually very resistant to antibiotics. Phages can penetrate and disrupt these biofilms more effectively. Looking at the future prospects, phage therapy has enormous potential. Firstly, personalized medicine. Since phages are highly specific, therapies can be tailored to individual patients. Phage banks may be developed to quickly match phages with infections. Secondly, we have synthetic and engineered phages, including CRISPR-enhanced phages, which can be designed to increase efficiency and target resistant genes. Thirdly, we have expanded clinical applications not only for chronic infections but potentially in cancer therapy and other advanced treatment. In conclusion, phage therapy represents a promising and innovative strategy to combat antibiotic-resistant bacteria. While more clinical trials and regulatory approvals are needed, it may become a powerful complement or even alternative to antibiotics in the near future. Thank you.
Any questions? Okay, thank you.
Thank you, sir.
Now another Samiksha. Be ready.
Sir, she had done with me jointly.
So we
It
are
is
done
jointly. Okay.
Yes, sir.
Yeah. Next, another Minch.
Yes, sir. Sir, I have shared my presentation in the group. Could you please share it?
Can you directly share it?
Okay, sir. Let me try.
Yeah. Yes, you can share it.
Okay, sir. Sir, are you able to see the screen? Hello? Hello?
Yes, please go ahead.
Sir, are you able to see my screen?
Yes, yes.
Okay, sir. Good morning, everyone. I am Lynette Minch from North Bengal St. Xavier's College, and I'm here to present my poster entitled as Oncolytic Virus Revolutionizing Cancer Therapy with Nature's Own Killers. Cancer is caused by uncontrollable cell division and is one of the leading causes of death worldwide. Traditional treatments like chemotherapy and radiotherapy can also damage healthy cells and cause side effects. Oncolytic viruses are specially modified or naturally occurring viruses that selectively infect and kill cancer cells without harming normal cells. They work by entering tumor cells, multiplying inside them, and causing the cells to burst, also known as lysis. This also releases tumor antigens, which helps activate the immune system to attack remaining cancer cells. The poster also explains how these viruses are engineered. Scientists select a virus, genetically modify it to improve safety and selectivity, and sometimes arm it with therapeutic genes to enhance anti-tumor effects. Some oncology viruses are already approved worldwide, such as T-VAC for melanoma and Dilatact in Japan for glioblastoma. There are many advantages of this therapy. It specifically targets cancer cells, has fewer systemic side effects compared to chemotherapy, stimulates long-term anti-cancer immunity, and can even reach metastatic cancer cells. However, there are challenges like immune system clearance, safety concerns, and human variability. Future research focuses on genetic engineering combination therapies with immunotherapy or chemotherapy. In conclusion, oncolytic viral therapy uh, Hello. Hello.
Yes, please go ahead.
Sorry, sir, I lost connection.
You can conclude.
Okay. In conclusion, oncolytic viral therapy represents a promising and innovative approach.
Okay, thank you.
Thank you, sir.
Any question from participants or judges? Okay, what is the role of natural killer cells?
Sir, could you please?
Okay. So what is the role of natural killer cells along with these oncolytic virus? Can you compare these two?
Yeah. Basically, nature's killer cells are lymphocytes that destroy virus-infected cells and tumor cells.
Okay. Thank you. Next
Thank you.
participant. Any participant?
Sir, Azna Shabi.
Okay, you can start your presentation.
Okay, sir. I'm audible, sir.
Yes, you are audible.
Good morning, everyone. My name is Azna Shabi from the Department of Biochemistry, SK Purval College, Kamptee. Today, I am going to present on artificial intelligence in bacterial infection diagnosis, treatment, optimization, and outbreak prediction. In this presentation, I will explain how artificial intelligence is helping doctors diagnose infection faster, choose better treatments, and even predict disease outbreaks. This slide indicates background and problem statement. Bacterial infections are still a major health problem worldwide. They cause many deaths and increase hospital cost. Another serious issue is antimicrobial resistance. This means bacteria are becoming resistant to antibiotics, making treatment difficult. Traditional diagnostic methods like culture tests take a long time, sometimes 24 hours to 72 hours. Because of this delay, patient may receive the wrong antibiotics, which can worsen their condition and increase resistance. So we need faster and smarter solution, and this is where AI helps. Artificial intelligence means computer system that can perform tasks normally require human intelligence. Machine learning learns pattern from data. Deep learning uses advanced neural network for complex analysis like images. AI can analyze electronic health records, lab reports, medical images, and genome sequencing data. AI does not replace doctors, it supports them by analyzing large amount of data quickly and accurately. AI helps in identifying bacteria faster. Machine learning models like random forest and support vector machine analyze lab data to detect pathogens. AI can also analyze gram stain images, microscopy images, MALDI, TOF spectra. When combined with whole genome sequencing, AI can identify type of bacteria, virulence factor, and resistance genes. This makes diagnosis faster and more accurate. Deep learning uses CNN to analyze medical images. For example, AI can detect bacterial pneumonia in chest x-rays with accuracy similar to radiologists. It also helps measure disease severity, reduce diagnostic delay, and standardize reporting. This is especially useful in hospitals with limited specialists. AI can predict whether bacteria are resistant to certain antibiotics by analyzing genomic data. AI can detect resistant genes. It can also estimate the minimum inhibitory concentration, which helps doctors choose the correct antibiotic dose. AI-based surveillance system can identify new resistance patterns early, helping prevent larger problems. AI helps doctor choose the best antibiotic based on patient condition, local resistance patterns, and lab reports. Clinical decision support system provides suggestions during treatment. AI also supports antimicrobial stewardship by recommending dose adjustment and suggesting when to stop or change antibiotics. This improves patient outcomes and reduces resistance. This slide shows how AI integrates different types of data. It collects hospital admission data, symptoms, lab results, GIS, location data. The AI model analyzes all this information together. Then it predicts possible infection cases and provides clinical guidance. This integrated system supports diagnosis, treatment, and outbreak monitoring in real time. AI helps detect outbreaks early. It monitors sudden increase in hospital admission, lab positive cases, geographic pattern. Using time series forecasting and geospatial model, AI can predict how infections may spread. This helps public health authority respond quickly and control outbreak effectively. Despite many advantages, AI has some challenges. First, data quality issues. Incomplete or biased data can affect accuracy. Second, some AI models are black boxes, meaning doctors may not understand how decisions are made. There are also concerns about patient privacy, data security, and ethical use. Additionally, implementing AI system in hospital requires training, cost, and technical support. To conclude, AI is a powerful decision support tool. It improves diagnostic speed, treatment accuracy, outbreak preparedness. However, proper validation, ethical governance, and collaboration are necessary. In the future, combining imaging, genomic, and clinical data through multimodal AI will further improve healthcare. AI will not replace microbiologists or doctors. It will work together with them to provide safer and better patient care. Thank you so much.
Any question from participants? Okay, I have question.
Yes, sir.
Uh, what is the interface at which AI will help the clinicians in identifying the pathogenic bacteria or organisms?
Sir, see, mostly MALDI, TOF spectra is used for bacterial identification.
Which spectra?
MADI,
Okay.
matrix assisted laser desorption ionization, time of flight.
Okay. So how will you correlate this data with the antibiotic resistance of particular organism, and how AI will predict which particular organism is pathogenic or not? Or how this data will assist a clinician or a microbiologist to discern the fact whether which particular the microbiota is having prevalence of which particular type of antibiotic resistance.
So one question from my side.
Yes, sir.
It is a theoretical or it is an hypothetically?
It is a theoretical based.
You are doing this activity in your lab?
No, sir.
Then how?
Sir, this presentation is based on theories, and
it is an hypothetical, na? It is hypothetical. If you are using the AI pictures and all these things, we can really it can use in the bacterial diagnosis. What are the parameters for different types of the bacteria? There is only one single bacteria which is responsible for the infections. Are you getting my question?
yes, sir. So detect and identify the single bacteria responsible for infection.
Yeah, definitely. But there are so many bacteria.
Yes, sir.
Their parameters also different.
Yes, sir.
How this particularly the AI which can help in the bacterial diagnosis infection?
Even identification of bacteria does not suggest whether this particular organism will be pathogenic or not, what kind of antibiotic resistance
Definitely,
is
sir.
having, what kind of the mutation is there or not. So
Yeah, yeah.
obviously one all this raw data has to be obtained through the laboratory work only. So AI, I don't know how that interface, how it will communicate with that.
Yes, sir. Sorry, sir.
No. You are not sorry. For what? You are giving a presentation. These are the questions
These
that
are the
will
ideas from your
arise in the
brain.
minds of microbiologists, and how to integrate that with the AI. So that will be the question. So you have to pose that question. You have to see what kind of discussion we can have in the area of microbiology, because obviously people are pushing the AI in everywhere, and the AI word is getting popularized, but people do not know how to use AI in any particular area. So it is the one should have curiosity, one should have question, one should discuss what best use we can have for that. But we cannot negate the dedicated work of microbiologist, because the basic data only the microbiologist will generate, and AI is the secondary. So it
Okay,
was
sir.
a nice presentation, but you should
Yes.
relate it with the experimental data only.
Okay, sir.
Okay, thank you.
Thank you so much,
Thank
sir.
you very much. Next participant.
Dr. Sneha Jaiswal, DB Science College, Gondia.
Yes, sir. So just a moment, I am just converting it into PDF file because I'm using my mobile and I'm unable to open it in PPT format.
Okay, okay, okay.
Just a moment, sir.
Jaiswal madam, any problem?
Sir, network issue is there and I'm unable to convert it into PDF.
Okay.
So,
Madam, you can
one
share your
presentation.
PPT in the group.
Okay, okay, sir. I'll be sharing it. Yes.
Sure. By that time, we can take another participant.
Yeah, definitely, sir. Srushti Mishra. Srishti Mishra.
Yes, sir.
Please share your content.
Yes, sir. So just a second.
Yeah. Hello.
Sir, are
अरे
you
कोणी
able to
तुझं
see
नाव
my
आलं तर
screen?
इतक्या रेडी नव्हते त्यावेळेस. बरं.
Sir? Hello? Hello?
Hello.
Sir, are you able to see my screen?
No, it is not coming.
Sorry. So now?
Not coming. Yes.
So now is it visible?
Yes. Be continue.
Okay, sir. Okay, sir. Good afternoon, everyone. Today I'm presenting my research on my topic antivirulence potential of betel leaf extract on pathogenic microorganisms. So basically, betel leaf, scientifically known as Piper betel, is widely used in traditional medicine and is known for its antimicrobial and medicinal properties. It contains several phytochemicals such as phenols, flavonoids, and essential oils, which may reduce the harmful activity of pathogenic microorganisms. Instead of killing bacteria directly, antivirulence agents work by reducing the ability of pathogen to cause disease. They interfere with virulence factors such as toxin production, adhesion, and biofilm formation. My objective for the research is the main objective of the research uh, is that I have to evaluate the effect of betel leaf extract on virulence factor of selected microorganism, which is Staphylococcus aureus in my research. Second is to assess the ability of pathogen to produce coagulase and determine whether betel leaf extract can inhibit the activity. So my hypothesis is that in this study, that is betel leaf extract has an inhibitory effect on pathogenic microorganism, and I assume that the bioactive compound present in betel leaf can reduce the virulence of the bacteria, especially their ability to produce enzymes such as coagulase. Uh, my methodology for this research is that I prepared betel leaf extract using ethanolic extraction method. Fresh betel leaves were collected and dried, washed in distilled water, and then dried, and were coarsely crushed in mortar and pestle with mortar and pestle. After that, ethanolic extraction method was used to prepare the extract. The extract was then filtered and was used for the further study. So, to evaluate the antivirulence activity, we performed the tube plasma coagulation test using the bacterium Staphylococcus aureus. So, basically, two tubes containing human saturated plasma were prepared, in which the tube 1 plasma with bacterial culture and betel leaf extract was present, and in tube 2, which was the control tube, in that plasma with bacterial culture but without betel leaf extract was present. Both tubes were incubated at 37 degrees Celsius for 24 hours to observe clot formation.
The presence or absence of clotting indicates whether the coagulase enzyme is active or not. So, as you can see my results, the result of my experiment showed a clear difference between the two tubes. In tube 1, which contained betel leaf extract, there was no clot formation in the plasma. This indicates that the extract inhibited the activity of coagulase enzyme produced by the bacteria. However, in tube 2, which did not contain the plant extract, clearly the clot formation was observed. So my observation shows that the betel leaf extract interferes with the virulence mechanism of bacteria rather than simply killing it. So my conclusion for my research would be that the extract inhibited the coagulase enzyme responsible for plasma clotting, which is an important virulence factor for bacterial infection. This indicates that natural plant extracts such as betel leaf may serve as potential alternative strategy to control pathogenic bacteria. Such antivirulence approaches may reduce the reliance on antibiotics and contribute to combating antibiotic resistance because betel leaf are easily available, require simple extraction method, and contain natural antimicrobial compounds. A drug developed from yes, sir. Piper beetle can be produced at a lower cost compared to many synthetic antibiotics. Thank you. Hello?
Any questions?
I have one suggestion, and that is for all the participants who are showing poster. Basically, it is really difficult to see the data or the presentation properly if you are making one single poster as a file, because we cannot read or we cannot understand. Visually it is not appealing. So it is my advice for future conferences or seminars. If you are making a poster, even though you are presenting, you should prepare as a single slides in a proper PPT manners.
Okay, sir.
Okay. Any question from the participants or judges?
So from my sides, one question. So are you extracting in the distilled water or it required the organic solvents?
Sir, it required organic solvent. I used ethanol for that.
But ethanol having its own some activity on pathogenic microorganism.
Yes, sir, it have some activity, but I extracted it till the ethanol was totally consumed or evaporated. After that only I used the particular extract. I have mixed the coarse powder into the ethanol, and after that, after filtering it, I have put it in boiling water. After its evaporation, I have used the extract.
Okay, when you are extracting one or the component, okay, what will be the temperature?
So temperature I have used around 60 to 70 degrees Celsius in boiling water bath, since ethanol is an type of alcohol which can even cause fire and is not preferred on high temperature. So I have used temperature between 60 to 70 degrees.
But if it is containing the certain volatile components, like the volatile esters are there, volatile oils are there, so at that time it can get evaporated one. So you can't get the proper activity.
Yes, sir, but
the
I
cold
wanted
maceration is the best one. The cold maceration is the best technique.
Okay. Okay, sir.
Okay, thank you for your presentation.
Thank you, sir.
Okay. So next participant, Dr. Sneha Jaiswal, DB Science College, Gondia.
Sir, I have shared my PPT in the group itself. Can you please
Okay.
share it?
Okay.
Yes.
Yeah.
Yes.
Please start.
So, a very good noon to everybody, judges. I am Dr. Sneha Jaiswal from DB Science College, Gondia, presenting the topic, very basic research we did. The bacteriocin producing Staphylococcus epidermidis, a novel antimicrobial strategy against MRSA, methicillin resistant Staphylococcus aureus. Next, please. So we all know that bacteriocins are the peptides or proteins produced by bacteria and are able to kill closely related strains at very low concentrations. And it is estimated that 99% of bacterial strains produce at least one bacteriocin. And we have Bactibase or database which integrates the sequences and physicochemical parameters and all the information about the structure, taxonomy, about the bacteriocins. Next, please. So in general, sir, next, please. There are 4 types of bacteriocins, 4 classes of bacteriocins, class 1, class 2, class 3, and class 4. Sir, next. So the problem. So methicillin resistant Staphylococcus aureus, it causes minor skin and soft tissue infection. Also, it causes life-threatening pneumonia and toxinosis such as toxic shock syndrome, everything. So it is very difficult to treat with antibiotics and hence become a problem worldwide in healthcare environments and community. So it requires effective measures for their treatment and spread. So Staphylococcin, the bacteriocins of Staphylococci, which kills human and animal staphylococcal pathogens, which are known to kill. So they can act as potential biotechnological applications in the treatment of staphylococcal infections, especially caused by the resistant species. So next, please. So bacteriocin can act as alternative to antibiotics. So in our study, we isolated Staphylococcus epidermidis from skin. So it is known to produce epidermicin, epidermin, PEP5, epilancin, K7. So such kind of bacteriocins are known to be produced by the skin microflora. That too, Staphylococcus epidermidis. And the mechanism of action of these bacteriocins is by pore formation on the bacterial cell membranes of target cells. Next, please. So with the preview of this, the objectives of our work are isolation of MRSA from pus sample, human pus sample, and isolation of S. epidermidis from human skin, and then screening of bacteriocin producers by spot inoculation method, and then to study the inhibitory activity of bacteriocin producing S. epidermidis by agar-well diffusion method. Next, please. So first of all, the human pus sample was obtained from the pathology laboratory in our city. And then that sample was streaked on mannitol salt agar. And after that, typical colonies of mannitol salt agar, they were picked. And the colonies were subjected for the resistance to the methicillin, cefoxitin, and vancomycin. And the colonies which showed, sir, next, please. Sir, next. Sir, next वो result वाला और एक बार हाँ yes, previous one. Yeah, so here how MRSA was isolated, we didn't do any PCR or something. We just we used this technique cephositin. If the zone is less than 14 mm, then we can say it is MRSA. And also we got a very less zone in vancomycin. There was no zone in methicillin, penicillin. Okay, so based on this, we concluded that the isolated staphylococci were methicillin resistant one, and we did coagulase test also, which was positive. Sir, previous one. The methodology. One more. Previous one. Yes, yes. So what we did, we took a swab sample from the human skin, like on the hand, forearms, and we grew it on mannitol salt agar, and the white colonies of S. epidermidis were obtained. And after that, that 5 ml culture, the broth culture were prepared, and 5 ml of overnight growth culture was centrifuged for 5 minutes at 10,000 RPM. And the lawn was prepared using the isolated MRSA, means the MRSA strain we isolated. The lawn was prepared using that, and well were prepared, and 50 microliter of this cell-free supernatant of epidermidis was put in the wells and incubated the plates at 37 degrees Celsius for 24 hours. And we observed the zone. Sir, next, please. So, next. So, these are the biochemical characteristics of isolated bacteria. This is from MRSA, and epidermidis from skin. We have 5 isolates. Next, please. Next, yes. So this is really blurred, but you can see here some zones of the supernatant. So this clearly indicates that. So next, please. The isolate one, the Staphylococcus epidermidis, the supernatant showed 24 mm of zone, and the pellet, corresponding pellet, showed only 12 mm of zone. So the supernatant showed more diameter as compared to pellet. So it clearly states that the bacteriocins are extracellular. Next, please. So, several strains of epidermidis isolated from the skin, they showed inhibitory activity against MRSA, which we isolate from pus sample. And the inhibitory effect of various concentration of bacteriocin, it needs to be studied. Also, we need to purify the active bacteriocin, and the broad clinical application is only done after standardization of drug formulation, either alone or in combination with other antibiotics. So hence, nevertheless, this bacteriocin producing strains of S. epidermidis, which we isolated, might be useful against MRSA infections after further investigations. Next, please. Sir, these are various studies related to the present work. Thank you.
Yes, any questions? Madam, I have one question.
Yes, sir. Yes, sir. Please.
When you are pick up the pus sample for the isolation purpose.
Yes, sir.
Every time you get the Staphylococcus epidermis, or there should be a presence of former Staphylococcus aureus?
Sir, we isolated Staphylococcus aureus from the pus sample, and epidermidis we isolated from skin sample, normal healthy skin. Two different sources.
The pus containing
are-
also the Staphylococcus epidermis also there.
Pardon, sir?
Both these microorganisms present in the pus.
Yes, they may be, but our focus was on MRSA. We want to get MRSA from the pus sample, and we want to use normal flora epidermidis from skin and see the interaction of these two.
Okay, when you are using the standard antibiotics.
Yes, sir.
Okay, so what will be the concentration of that antibiotic?
Maybe, sir, I have to check it. Maybe 30 microgram. There are different concentrations of antibiotic we used. Cefotoxime was 30 grams.
Okay, 30
was,
grams or 30
standard
micrograms?
Micrograms, sorry.
Okay.
And here we didn't concentrated or quantified. We just took 50 microliter of supernatant.
Okay.
Of the extract of that epidermidis.
Okay.
Yes, sir.
Thank you, madam, for your nice presentation.
Thank you, sir. Thank you so much.
Okay, so next participant, Mr. Vivek Jede, Assistant Professor.
Yes, sir. Yes, sir. I am audible, sir.
Yeah. Please share your PPT, sir.
Yes. Is it visible, sir?
Just yes. Please in a Zoom.
Okay. Just 1 minute, sir. Okay. Myself, Vivek Jede. My topic presentation is new molecular biology approach for authentication of medicinal plant powders. As you all know that medicinal plants are a very important medicinal properties, and so different kind of formulations are used in pharmaceuticals as well as in science research, along with different kinds of powders, tablet, capsules can be performed for a pharmaceutical application as well as for research purpose. But the problem is that when we authenticate that particular sample, if sample is there, like a powder sample is there, and we want to authenticate it, then there is a problem with authentication because powder do not have any morphological features. Morphological features are lost, microscopic in analysis are not available, and phytochemical similarities are the major cause with all the powders. All the powders shows some phytochemical similarities, and adulteration is another problem with commercial powders. So to sort out this problem, we took an approach that is DNA barcoding approach. We find out many research papers where they use DNA barcoding for authentication of such kind of powder from medicinal plants. In that, two common methods are there. That is the RBCL gene sequencing and ITS region sequencing. In my research, I have 3 plants, that is Moringa oleifera, Berberis aristata, and Calothrunus rosas. From these 3, we are facing much problem with the Berberis aristata, because Berberis aristata is the plant which is actually Himalayan plant, and it is annual plant. So during winter season, it is not available, and because of the region, Himalayan region is far from the Maharashtra, we cannot directly reach to the particular that plant. So we purchase it from authentic herbal vendors, but we have to assure that the powder should have the all the properties of phytochemical properties. So we go for this, that is the genomic approach. And in here we extracted genome from that powder, PCR amplification, Sanger analysis, and sequence analysis. In this way, the powder is converted into using molecular technique. It is converted into identification purpose. Here is the RBCL sequencing of Moringa plant, which I use for my study. It is the RBCL sequencing of Berberis aristata. Actually, we use root of this Berberis aristata for our research, that is nanoparticle synthesis. And for that, as morphological identification is not possible, we go for a molecular analysis. This is the sequence for my third plant, and you will easily find here that the percentage identity of my sample is 99.92 with the present data with the data which is present in internet, and from that we can easily get the phylogenetic tree also, to which authenticate our research. So from our research, we can conclude that successful authentication of the molecular way of the medicinal plant powders can be possible, and with blast analysis, 98 to 99% similarity is acceptable for any Q1 or Q2 research journals. What is the importance of molecular authentication? That is, it does not raise any kind of question in your basic research. It also assures the quality control, standardization, and research reproducibility. Conclusion, that is DNA barcoding represents reliable, objective tool for authentication of medicinal plant powders. And it is also best approach to hold the promised future research in phytochemistry as well as in nanobiotechnology. These are my references, and thank you.
Thank you very much. It was nice data that you have presented. I have one question.
Yes, sir.
Because for a molecular, assessment of the DNA,
yes,
or for barcoding,
yes, sir,
the biggest issue is the probes, right?
Yes, sir. Yes, sir. Yes, sir.
So how one has to go? What are the approaches you suggest for going for this kind of molecular diagnostic?
Actually, sir, when we go for that, actually we are facing real issue when we presented our data of Berberis aristata. So for that, we take help of this company also, that is Green Array, and we togetherly develop a strategy how to identify that particular powder sample, because there are a lot of standard markers are available, but for the plant there are limited forward as well as reverse primers are available. So we use the reverse and forward primers which are actually used before plant identification, and that data is easily available in during blast analysis, sir.
Okay.
So we use only standard methods, sir.
Okay, fine. So you corroborated your data with the earlier presented data.
Yes, sir.
With a-
I compared it with earlier data, and we get the phylogenetic tree, sir, as shown in this PPT. And that will acceptable for Q1, Q2 journals because they want authentication, not not with morphological identification. They also want molecular identification.
Molecular identification.
Yes,
Right.
yes, sir. And it is much enough for their to accept our study, sir.
So this
In biotechnology also, sir.
this data is published,
Yeah,
or
sir,
it is
we are.
unpublished data.
Yes, yes. No, sir. Actually, it is unpublished data. We have submitted for 2 journals, and they accepted our, and it is in actually reviewing process, sir.
Okay. Thank you very much.
Thank you, sir. Thank you. Thank you so much, sir.
Next participant. Who is the next presentation? Anyone from the participant whose presentation is ready, he can join for participation.
Hello, sir. Can I represent my presentation?
Yes, yes, please go ahead.
Okay, sir. I am sharing my poster. Is my poster visible?
Yes, visible. Go
Okay,
ahead.
sir. Good afternoon, sir. My name is Amargiri. Today I am going to present my research on producing bioethanol from potato peel waste and newspaper waste. As we know, fossil fuel like petrol and diesel are limited and they also cause environmental pollution. At the same time, large amount of kitchen waste like potato peels and paper waste like newspapers are thrown away every day. These wastes increases landfills problems and environmental damage. So I asked a simple question: can we convert this waste into something useful? The answer is yes, and that useful product is bioethanol, a clean and renewable fuel. The main aim of my research was to use potato peels and newspaper waste as a raw material to convert them into sugars to produce ethanol using yeast and to test the quality of the ethanol produced. Why potato peel and newspaper? Potato peels are rich in starch, newspaper are rich in cellulose. Both starch and cellulose can be broken down into simple sugars. These sugars can act as a food for yeast. In this study, we use Saccharomyces cerevisiae, which is commonly known as baker yeast. It converts sugar into ethanol through fermentation. My methodology was first we collected potato peels and newspaper samples. The sample were dried and powdered. Acid hydrolysis was performed to break down starch and cellulose into sugars. The solution was neutralized to maintain proper pH. Yeast was added to prepare the solution. Fermentation was carried out at 28 degrees Celsius for 7 days at dark room. After fermentation, ethanol was confirmed by different tests such as alcohol indicator test, flame test, pH test, refractive index test. The result, the combination of potato peel and waste newspaper gave the maximum best result. This shows that combining starch rich and cellulose rich waste products improve fermentation and ethanol production. Conclusion: from this research, we can conclude that waste material like potato peels and newspaper can be converted into bioethanol successfully. This method is eco-friendly and low cost. It reduces waste disposal problems. It supports renewable energy production. Therefore, bioethanol production from potato peel waste and newspaper waste is sustainable and practical solution for future energy needs. Future scope is in this if this process can be scaled up at industrial level, it can help to reduce landfill waste, lower environmental pollution, and contribute to our green energy development. Instead of seeing waste as a waste, we should see them like a resource. After adding some biological process, we can turn them into wealth. Thank you.
Thank you for presentation.
Yes, sir.
I have one question.
Yes, sir.
So you did bioethanol production from potato peel waste, right?
Yes, sir. Newspaper waste.
So whether you have estimated the production quantitatively or qualitatively?
Quantitatively, it was 54% in 100 ml solution.
54% of what?
Alcohol in 100 ml of solution. And this
No,
is
I'm
my
asking.
ongoing research, and I am trying to make an applicable from this alcohol.
No, my question is 54% of what? What was the
Alcohol.
volume of
Volume.
Which component?
Ethanol.
So what was the volume of your waste? Whether it was solid, semi-solid or liquid?
First, I taken potato peel and I made it in powder form. Then I made a media from it by using baker yeast. So it was in liquid state.
Okay, so you have used these potato peel waste as a source for carbohydrate, right?
It was, yes, to convert them into sugar. It was rich in starch and newspaper was rich in cellulose.
Mhm. Okay, fine. Thank you.
Yeah, thank you, sir.
Next participant.
So can I present
Yes,
and share my screen, sir?
yes, please share.
Sir, if our presentation is over, can we leave the session?
Yes, you can leave the session.
Yes, sir. Thank you.
So is it visible?
Yes. Yes. Yes.
Yes, yes, visible.
Good afternoon, everyone. My name is Shrimoy Sengupta. I'm a PG student from Department of Botany, BHU, and it's a very last-minute presentation that I'm giving here, but I hope that the idea behind it will be impressive enough. So my poster is titled "Review on the Quorum Quenching Ability of Phytochemicals of Northeast Indian Plants as an Alternative to Antibiotics." So now let's start with the problem we are all familiar with, which is antibiotic resistance. Now, according to WHO, it is one of the biggest threats to global health today. As we can see in this poster, in this picture, resistance is driven by factors like overprescribing of drugs or patients not finishing the given course treatment duration or overuse of antibiotics in agriculture, which ultimately leaches into the crops that we eat. Now, the traditional antibiotic approach is a search and destroy mission, which means that it kills bacteria. Once it finds the bacteria, it kills bacteria. But this process creates a massive evolutionary pressure as well. The bacteria that are able to survive, they mutate and then they become what we call superbugs. Now, because of this, increase in resistant strains, there is increase in morbidity and mortality as well all over the globe. Now, because of this, scientists are desperately searching for alternatives. We need a way to treat diseases without actually causing the bacteria to develop resistance. Now, the answer to this, I believe, is that it is not killing the bacteria. Rather, we must simply stop them from multiplying within the body. And then the immune system can take care of the rest and flush it out. So now let us talk about quorum sensing. What is quorum sensing? Now, quorum sensing is a process through which bacteria communicate with each other. Now, bacteria aren't just solitary cells. They are social beings and they communicate with one another. As we can see in this diagram, at low cell density, they will have individual behaviors. But as they multiply, they release chemical molecules, signaling molecules called autoinducers. Now, when these autoinducers, they reach a critical concentration or a quorum, they trigger a group-wide change in gene expression. Suddenly, the bacteria, they stop behaving as individuals and they start acting in a coordinated manner like an army. They form impenetrable coats around them called biofilms. They release toxins. There is expression of various virulence factors. So bacteria specifically, they only attack the human body once they reach a concentration where they can safely multiply without being destroyed by the immune system. Now, as we can understand that their strength lies in their communication. So our strategy should be to cut off this communication process, and that is what is known as quorum quenching. If we look at this diagram, there are three primary ways in which quorum quenching can be processed. First is we can stop the bacteria from synthesizing the autoinducer molecules in the first place. Second, we can destroy or inactivate signals once they have been released and they are going to the other cells. And third, we can block the receptors from receiving the signal itself so that further gene transduction does not happen. Now, what tools do we have to achieve this? The answer is phytochemicals. What are phytochemicals? They are natural compounds extracted from plants. Now, plants, they have been fighting off bacterial infections for millions of years. They don't need antibiotics. They have inbuilt mechanisms through which they fight bacterial infections. Now, in quorum quenching, plant extracts, they act as inhibitors. They can interrupt the pathways, degrade the autoinducers, or interfere with the receptors as well. Thereby, they effectively silence the bacterial communication or quorum sensing. Now, this brings me to an important question. Why do we choose Northeast India for this? Where do we find the best phytochemicals for this job? Now, Northeast India, it is recognized as a global biodiversity hotspot. What does this mean? That this region alone harbors 50% of India's total plant diversity, and roughly 40% of its species are endemic. And what does endemic mean? That they cannot be found anywhere else on earth. But this is not just about biodiversity. It is also about traditional knowledge. There are various indigenous communities, tribes in Northeast India that have been using these phytochemicals as a repository for their own ethnomedicinal practices. Now, these communities, they have used these local plants for traditional healthcare, which gives us a target point or a starting point for our modern development of pharmaceuticals. Now, the scientific community is already validating this point as well. Currently, over 20 plus ethnomedicinal plants from Northeast region have been evaluated for their biofilm inhibition properties, and pyocyanin, which is a pigment, a virulence factor in Pseudomonas aeruginosa, inhibition of that has also been checked as sub-MIC levels. We have also seen studies where the quorum quenching activity using Chromobacterium violaceum as a reporter strain has been used, and it has and several plants have been identified that interrupt the AHL, which is acyl homoceran lactone, which is used by C. violaceum as autoinducer molecule. Now, there are also research which highlights that plant extracts from this region, Northeast India, such as Curcuma longa, they significantly reduce the production of EPS, or the coat that helps bacteria protect themselves and helps in the formation of biofilm. So these plant extracts, they reduce the production of this EPS. Now, to conclude, I want to emphasize that there is fundamental difference between this approach, the phytochemical approach, and standard medicine. Now, unlike antibiotics, which kill bacteria and create a selective pressure, quorum quenching just disarms the bacteria. It is not able to communicate with one another. It inhibits the production of virulence factors, inhibits the production of biofilm, without which the bacteria are neutralized. But there is no evolutionary pressure on them to mutate or produce other strains. Now, these compounds derived from medicinal plants of Northeast India, they are generally safe for human use because they are using nature's own defense. Although none of the plants have yet gone to human trials or drug trial levels, but current work is being done to extract the phytochemicals in such a way that at low concentrations they can be used for treatment of human diseases. Furthermore, these phytochemicals, they are also broad spectrum, which means they kill gram negative and gram positive irrespective of their genus or their species. Now, the biodiversity of Northeast India, it is area where we can work upon, because there is a lot of possibility of extraction of compounds from this region, which can help in the future for development of phytochemicals and reduce our dependence on antibiotics. Thank you so much.
Thank you. It was a nice presentation. So the difference between quorum sensing and quorum quenching is the inhibition of the signals which these microorganisms give or spread to the community for a buildup of various superstructures.
Yes,
and one
sir.
of the superstructure is a biofilm.
Yes, sir.
You mentioned the Curcuma longa.
Yes.
And why it is Northeast specific? Curcuma longa is pan-world present medicinal or the spice.
Yes,
isn't it?
sir.
So what is the specific about Northeast India regarding Curcuma longa?
So, Curcuma longa, it is one of the. So now there is a region in Meghalaya, in Jaintia Hills, and the most famous variety of Curcuma longa in this region, it is known as, I think, Lagdong turmeric. Now, this is the normal Curcuma longa. It has a the amount of curcumin in that is low, whereas the variety found in these hills of Meghalaya, the amount of curcumin in them is high. So there is a higher probability of extraction of that compound from these Curcuma longa plants found in that area.
This is altogether different aspect. You are talking about the concentration. You can take a pure curcumin that is available in the market and you can dosage it. So it is not about that. That's a agriculture point of view, or that's a extraction point of view. It is nothing to do with the quorum sensing or quenching, right? You have to focus on the how the curcumin is causing any effect on the sensing or quenching, right? That should be the question you should address here. Or any particular mechanism which helps in quorum quenching.
I mean, I have not searched on it a lot, but I have this idea that as I was reading, I found that the high levels of curcumin make them more effective at silencing bacteria. That is what, while I was reading that particular paper, the particular paper that I was reading, I found. But for sure, I will research more on this as to why the curcuma longa from Northeast is, why specifically we should use it from there or not the other places.
Okay, thank you.
Thank you, sir.
Next participant.
No one is there, sir.
All participants are,
Over.
presentations
As
is
per
over.
my list, no participant right now.
Okay, okay. So shall we conclude?
Yes, sir, definitely.
So I will call upon all the participants and judges to open their video. Thank you very much, all the participants. You have presented the data very nicely. You have answered the queries of judges and participants. Dr. Banginwar sir was the host for this particular Zoom meeting. So thank you very much, Banginwar sir. He's one among us. Dr. Davande sir, Dr. R.U. Thakre sir, and all the organizers as well as participants for this national conference. We are concluding one of the important session, that was paper presentation, oral presentation, poster presentation. So thank you very much on behalf of the judges for this particular session. Now over to the host, Banginwar sir.
Thank you, sir. So as one of the important task during the conference in an online mode. So first of all, thank you to all the participants who share their views on the frontiers in life sciences and material sciences at the AI and IPR interface. So their ideas will be very nice. Presentation is very nice. And their ideas, reality. So you can convert your ideas into the certain technology. That is your future. So with this, Mark, I will conclude. And today's session is get over with the permission of our conveners. Thank you so much. Thank you, Gadpaile sir, for acting as the main role in today's session.
Thank you, sir. Thank you very much.
Okay, so meeting is get over. I will stop the streaming right now.